So welcome to Lab Practical 2 as a part of our course G07 on phylogenomics, where we're looking at linking metadata to large-scale phylogenies. So we're going to do this in RStudio as before, and we need to install certain packages such as ggtree to achieve this. What we're going to do with this package is then take our sequences we had before of those FMDV VP1 regions and convert them into a tree that we're going to store as the variable tree1. And then we're going to visualize a simple phylogeny using this ggtree function, which just takes in a tree-like structure, our tree1 here, and visualizes it using as a single function. So we get something like this at the end, a simple phylogeny. And we can build this up gradually by adding extra arguments to this command. So for example, if we add the command tiplab with no horizontal adjustment and a certain size, we can see a tree like this. So admittedly, these names we just downloaded are very, very long and need some parsing, but we can certainly see certain ones if we modify them in a way that suits. Similarly, there's also an option to align them, to right align them. So here, obviously, we need to adjust the margins of our plot, but at the same time, we can still see that they're all right aligned, and therefore sometimes that can be easier to read as well. You can see that we also adjusted the size parameters to what suits us too. Furthermore, we can use the tree scale to add in an automatic substitutions per site scale bar at the bottom here as well, which is very useful for our plot, and we don't need to place it using coordinates. We can also add a rate of substitutions bar at the bottom, going all the way through from the root, all the way through to the tips. So that gives us a better estimate of the root to tip distance for the different samples. Furthermore, we can add different parameters into the initial function ggtree to make it, say, color blue. We can change the line type to be, for example, dashed like this. We can change the sizes again to be smaller and bigger as suits our needs. We can add more things onto it. So we can add node points as well to our phylogeny. So you see there we've got the internal nodes are represented. And the same way we can also do it for the external nodes. So this is more conventional because that indicates the sampling points shown by the black nodes. And therefore we can map onto this different colors to identify particular clades that are of interest. We can also identify the nodes that are of interest, which is the first step in coloring different ones. So you can see that we can specify that we're looking at node 109 or node 106 or this node 129 using these nodes. We're going to look for that node 123 here, which is the most ancestral one down here. Well, arguably maybe 101 is more ancestral, but there's just a select a node to examine. So what we can do then is take that node and label it as the root. We're going to color it blue and have some offset. And we do the same with this clade label function. Using for node 129 as well, we're going to call that group outbreak and we're going to color it red. So what we'd see from that is there's these labels attached to these specimens where the offset changes where the labels are placed. We can also color these as well. So you can see that on the right, what we're using is the geom highlight function. For node 129, we'll color all the descends associated with that nose and fill it with red. So we can highlight certain clades that happen to be of interest. We can also collapse nodes. So you see this old tree here with that clade specified. You can see here it's actually being collapsed into a green shape using geom.2. So that takes that node, and we're highlighting it in green, but we've collapsed it using that collapse function. So we can reduce clades if that's also of interest for our hypothesis. Here we see that same node that same ancestral node, node 152, has been rotated, and then we've highlighted that the same way as before. So you see here we had this outgroup branch adjacent to this tree. We've now inverted the tree. They see the most distant branch here, that long one here at the edge has been rotated around. So we can rotate our nodes as well. We can also make the tree layout different, so we can make it circular if we want, instead of a phylogram. We can add the names as well, at an angle too. Of course, that works, doesn't work very well So what we can do then is create metadata that we're going to add onto this phylogeny. So for example, we might have 3 layers here. So creating a data frame where we have one column called first, a second column called second, a third one called third. And we're adding into the first one 33 A's, rep A 33 times, rep B 33 times, 33 B's, and 33 C's together, all in one vector together. Then we have a second one where we rep 1 22 times, rep 2 44 times, rep 3 33 times. So that sums to 99, corresponding to the first vector. But you see it's slightly different. And the third one, we're going to call it I, double I, and then triple I for 11, 77, and 11 again. So sums to 99 because we have 99 samples. And what we're adding into this data frame is the tip labels, our sample labels are added into that. And then we can view our data frame, which looks like this. So this is the metadata we're going to represent around our tree instead of having these labels, which don't really work. So if we do this using this function, we use geom new scale, we import that. We need ggplot2 as well. We're taking our data frame here along with our plot P1. We have an offset of 0.01, a width. This is for the circles that are in the plot. We determine the angle that works, the offset, and the color scheme, which is scale Verdis. What we should see then is something like this, where this contains our first, second, and third metadata. The colors reflect the values that are present. So you see there, we have 1, 2, and 3 for the first few colors. The second, ABC for the first few colors. The second shows 1, 2, and 3 in terms of their phylogenetic pattern. And then we see we have certain ones with the values of 3. There's just 11 of them in that case. And we see the other 11 here at the edges of certain parts. So this is what we can kind of do with ggtree, and we're going to switch then to our studio now to continue this. So we can check that we have ggtree installed. If you don't, you'll have to install it. We have our old NGS 6 distances 5, so we can check what's in our tree structure. We see that we have our 99 tips with 97 internal nodes containing the BP1 gene. So as mentioned, we can simply visualize that with the function ggtree on its own. So that's a simple way of drawing a tree. It may take a second to execute on your computer. And then we can add tip labels as well if we want. And we can change this to be a small size. Maybe that would accommodate them more effectively, but you can see it's tricky because you can't see much there. We can also align them to the right too. So you can see them there all together. And then we can add a scale bar to that as well, which is also useful. We can add one at the bottom again, if that's useful for us too. We can change our different line types. So we can change it to line type 3 and create our tree like that, although that's not the most clear visual ever. So there's lots of things we can modify inside here. So we turn that to red, for example. And we see a red tree. We can turn it to yellow, and we get a pretty awful tree. And then we can add the node tips here, and the external branch of the tip point as well. So you can add that in there. So you get both internal and external nodes are highlighted here. Obviously, that's very busy, so it's not really a wise thing to do, but certainly the external node is common to highlight them. We also can identify the node tips with this function here. So we're adding on the function to put them in there. We can see all the sizes again if we want to make them bigger or smaller. What we can do here is change the nodes that we're calling root. So for example, we might look at 101 instead, call that the root. So if you try that only with the blue coloring for the root, that's 101 is down here. We can see that we got the root specified, so that doesn't really work in this instance. We can try another clade number, I think 141 as well, could be worth trying. And see there we have our outbreak clade is labeled differently now. We can also highlight that. So we could highlight clade 141 if we wanted. So let's try that with geom highlight. So you see that one is colored, and we can keep adding those on if we want to. We can just build up our plot like that. 129, We're going to shade that green, and then maybe we could do 101 as well, and color that, let's say, gray. Let's see how that looks. There we have several 101. That's what I wanted. Maybe we need to change this. There we go. So we have different colors represented on this tree. We can collapse nodes. So let's say we're going to collapse 101 and then mark node 129. We can do that too. So you see 129 is present. If you kind of zoomed into this tree, you've actually collapsed the external, the molar divergence samples here. Equally, we could do that for 141 and still highlight 129. So we see that there. We could pipe this into collapsing multiple nodes and really reduce our tree further. And again, we see much less now, but that clade is highlighted, and we can continue to highlight that as well if we if we wanted to. So we really highlight that like that. So there's lots of things we can do. And then we can rotate around nodes. So might rotate around 101 and highlight that node. So you can see that's been rotated now. And then what we're going to do is store our GG tree as a circular tree. And we're going to call it P1. So if we look at P1, we can view it like that. So there's a lot of things we can add on there. If we add on the label tips as shown by the previous diagram, it's not going to be, even if we reduce the size, it's still not going to be very useful for us. So we can talk about that, but it really does work. So we encode the metadata table. So you can see what you can see is that this table, because we look at the structure of the tip labels, we have 99 entries inside there. So that's why we create a table with 99 entries. So you see for each one, first, second, third, we have 99 entries that are just made up data. But my point is you can layer on multiple data sets inside here. For example, we could have another one just called fourth with different symbols and values. And we can just keep adding those if we wanted to. What we need to do is install GG NewScale and ggplot to visualize this. And what this is doing is we're setting the parameters to determine the width of the plots that we see. We can change that. The colline's angle is presented there. The offset is presented there, and this is the coloring options. So if we put that all together, we'll get that visual here. So what we have to do is toggle the width to incorporate that fourth one. So that might be a push on these particular samples because of the lack of space here for the 4 rings. So we have those categories. So you see there X and Z is represented as well. And if you zoom in, we can see there's a fourth one there. So that kind of gives us an idea about how we can create different types of trees and visualize them effectively with GG tree. So let's move on to looking at DNDS ratios. So we'll move back to the slides. So now we're going to look at linking metadata to large-scale phylogenies. So again, this is with R, and again, we're using the APE package to compute DNDS ratios. We're going to use this our example data, the sequence.fasta file. We're going to read that into data1, check that we read it. Note that to compute DNDS ratios, we want our alignment to start with ATG and finish with a stop codon. And what we're going to do is specify that we want the unique sequences that are present going from bases 1 to 648, the length of our sequence, for these samples, because you kind of instances where they actually have the same sample name the same sequence for different samples. So then we're computing the DNDS ratios for this particular data set. We're going to ask for the details to be turned off, and that computes a pairwise matrix of DNDS ratios. So what we'd expect to get is something like this. So you can see their relative rates, and they're all quite small, as you would expect. What we can do then is take the elements in the NDS1, which are not numbers or are infinites because we have no synonymous changes, and set them to be equal to 1. And then we can view our matrix like that. In this case, we don't know NaN values or infinite values, hence that's not an issue. What we can do then is take the sum of the NDS values and divide it by the length of the number of entries that are present. And we can also do that to test the standard deviation as well. And what we get then is a DNDS ratio average one of 1.126, indicating that there's purifying selection as we'd expect. We also see that the standard deviation of the variation in estimates between the different samples is quite small as well. So there's only one sample pair for which we actually find a surprisingly high value compared to the rest, which is why it's good to plot that and visualize it effectively. So to explore this further, what we can do is, if we go back to our matrix, it's the value here between our sample 85 and 86. So we can then explore them further here by selecting those from the data matrix and getting more details from that. So we can see that we have at non-degenerate sites, we have 7 transitions, 5 transversions. At 2 degenerate sites, we have 4 transitions, 2 transversions. At 4-fold, we have got 2 and 2 of each. Now see that estimate for each of those is surprisingly high, but that's the values that we're given. So let's switch to our studio to explore that further. So here we have our sequences being read in. We check the structure of data 1 then to see if that worked okay. So you see our sequences are there. They're all 648 bases. And we're looking at 7 of them. So what we're doing is we're taking our DNA string set. We're getting the unique samples among these. So if we look at that, then we can see we have a list of 7 of them. We're looking at as a DNA bin. So we look at the structure of that element. We see we've got a list of 7. And we can kind of see more details there. The data. That's what we're trying to look at. So you can see there we have our list of 7 samples that we want to explore further. So what we can do then is create a DNDS ratio matrix, which you see there in DNDS1. So that's our pairwise matrix of the different values. If we turn the details to be true and quiet equals false, we just get more information produced there for each of the pairwise comparisons. So you can see there's a lot of changes here. And you see that the samples that we're trying to look were between samples 4 and 5. So scroll up to there. Those are the rates that we have, which don't appear to be quite different from the others. There's just fewer changes. So perhaps there's just fewer synonymous sites at that particular region. So you see that some of the other samples have quite high, comparatively higher divergence rates. So then here we can get the average across this matrix, which is 0.126, get the standard deviation as well, and we can get a histogram to explore that further, and then explore those ones where we have some sort of difference as well, just to see what information might be available too. So you can see that there. If we want to explore them further, we can go back up to our list, or is it our data? It's our values up here. We have our data 1. So we can see our alignment across here. So in our next video, we'll explore other aspects of alignment visualization approaches. So next in our practical here, what we're going to do is substitution model comparison, which we touched on previously. Here we're going to assess the models using model test ng on the server. So model test ng considers things like the number of models that you want to examine, the relative equality of the different nucleotide or indeed amino acid sequence, if invariant sites are present, the number of categories for models with great variation among sites, the criteria of interest, parameter estimation, and if we want to do model averaging. So you can see more at this particular website where it details more through the command line. So we've kind of these variety of models that are available. where you have different frequencies for the base frequencies. So you can see there the juice cantor is more simple. And we kind of examine many of these models and more beyond. So you got TN93 I mentioned before, K81. So you can see as there's quite a few different options. So here we're going to try to use a maximum likelihood approach rather than the default maximum parsimony approach. So we can toggle that when we were running our commands. So we have this table here, and that indicates the different criteria to decide which substitution model is most effective, and that's not trivial to do. So you can see there's three non-Bayesian approaches: the likelihood ratio test, the AK information criterion, and the corrected version of it. And we've got the Bayesian information criterion and decision theory again, our Bayesian approaches for that. So recall that what we're trying to think about when we compare two different models, we look at the likelihood of each different model, take a scale by its log. So just do a subtraction of one from the other. We take twice this value and then compare that to a chi-squared distribution to see if one value is more divergent. So if we compare these two models, where HKY is 85 versus GTR, if the difference here is very, very large, we can see that one model is better than the other. We have to consider the degrees of freedom, where there's a difference of 4 parameters between these two. And what we're saying is that the difference here does not exceed a threshold value, indicating a p-value of less than 0.05. Of course, that's not that's just another percentile. But it certainly indicates that the comparison of these two models shows that it's not an extreme difference between them. So here we're thinking that the GTR is a bit better, but it's not clearly better. So what you can do in the lab is copy over this file to your folder location model test-GUI, and you can also copy over the sequences that you need to your local folder, and run this program model test.GUI with a / and a dot full stop at the front to make sure it's executable in your folder. And a window will pop up after there. It's a GUI, so a window like this will appear. What you can do then is load our MSA, your seeks.phas, inside there. You can also install the built-in dataset tiny.phas as well, which is the one given here. It is 6 tasks and 631 sites. And what we can do then is modify the settings here. So these are all the settings. We're looking at DNA. We want to run model test versus say RAXML. These are the models that are possible. We can have an ascertainment bias correction. Rate variation here is there's a few options. We can change the number of categories present as well. We can assume that the frequencies are equal or maximum likelihood. So there's many different things we can tweak. So what we can do is load our MSA, toggle the settings, hit run, and you'll see this. So we'll say what type of signals you're examining, the number of models, the parameters for your different options that are present. And then at the end, it will show this is the score for the different models, and these are each of the models on a different line. This is the log likelihood. So what we can see is that the TPM1 plus invariant plus gamma 4 has the highest score and the lowest log likelihood, indicating that it may be the best model in this particular run. We can also consider the criteria across the BIC, AIC, and AICC. And what we see is that their log likelihoods here appear to be all the same, but you can see that the scores differ slightly. At the same time, they appear to be in agreement that this is the best model, I think. But that may not necessarily be always the case. So you can try that. When we try that for our VP1 FMD sequences, we can repeat that using the same parameters. What we see is that the TPM UF plus G3 is the best. This is a variation of the K81 model, allowing unequal base frequencies and 3 symmetries. as shown by there. And this is the kind of result that we get. You see the score is quite marginally ahead of the TIM1 plus invariant model. And we see there that there's the log likelihood estimates as well. So let's now go to the server to explore this further. So what we can do first of all is create a folder for our analysis. So we're going to create a folder here. I'm going to call it Phylogen. And what we're going to do is copy over those files that we mentioned earlier, which are samples. So the first one is model test GUI. So we're going to copy that to this folder and check that it's present. Oh, we have to go into that folder. Yeah, there we have it. The next one was seeks.phas. So again, we check that that is present. You can copy this straight over into the this portal, and then we're executing this. And what we need to do is click on this X here. That's the X11 window containing this tool. So you click on load MSA. We can select seeks.phas. We can toggle our settings that maybe we want to include more gamma categories for rate variation. That's for maybe that'll create more memory. We run that and let it run. So what we see is that as executed this model is saying that the use counter with invariant plus 11 gamma categories appears to have the maximum score and in this algorithm, although I'm not sure if it's best log likelihood. Yes, it appears to do so. Although we can see that the use counter with just without invariant is also very good. We see our results there. Sorry, the HKY plus invariant is the best. So you see our score there is best for that option. For the ARC, got TIM1 plus invariance, that differs. ARC with corrected, again, we get a different one that's the best. So you see HKY plus I is different. So this is the tricky part with the modeling criteria over second, which is the best model for our data here. What we can do is close that and copy over the sequences we were looking at before, sequences.fasta, or we could actually copy over other sequences in model test. So we're going to copy that over here. And those example data, we're going to copy that. We're going to copy that here, chana.phas, and then run the model test GUI again. So you can see there we have to load our chana.phas files. We see that 246 taxa across 4.4 kp, so a lot of side patterns. This might take a lot of memory. So let's see how this runs. In terms of the settings, I've no reason to suspect that there should be any difference from anything else. So we can see that this is going to take a bit of a longer time to execute, but it's running through fairly quickly. And what it's doing there is showing the different models that it's testing. I know we can go to the results. So that has indicated that the TIM2 plus invariant plus gamma 4 is the best based on the score for BIC. For AIC, we see the GTR plus invariant plus gamma. The TIM2 is 1 and second, whereas that's first and second is switched here for BIC. And AICC says the GTR 1 is the best. So that kind of gives us an idea that certainly these two models appear to be the best, and their scores appear to be somewhat better than the other ones based on our evaluation here. So we can then take this forward and then look at the tools that we're using and construct a tree using this substitution model, which will then give us a better sense that our phylogeny as constructed uses the best tools that are available for this. So for the final part of our project, we're going to look at tree construction with RaxML on the server. So RaxML is a very commonly used tool for phylogenetic reconstruction. It is uses maximal likelihood approaches as per the name. The most recent version of it is called RaxML NG. There's been other versions that have been used as well. And what this does is that it creates a tree, but does a series of subtree pruning and regrafting steps. So it takes bits off and moves them around and switches that to efficiently find the optimal model. It allows numerous different substitution models as shown by the list here. So once you've done model test NG, you can then run RaxML based on the substitution model that was most effective, and then visualize it with R. So the first step in this process is to check that the input sequences seq.phas is what we're going to use here, are valid for phylogenetic assessment. So we use this command here, RaxML ng --check. MSA is the multi-sequence alignment input, and then we take in seq.phas. The model is GTR plus gamma here, and then we have a prefix indicating what type of output files we want for the name. So that allows us to align these 648 base sequences, where we see that 71% of them are actually invariant. So that's given by here. And then we have those parameters I mentioned already. So what we can do next is actually run this with the model, assuming that our sequences are okay and are alignable. Often people make mistakes with the sample names. There's some weird symbols, or there's other errors in actually those sequences themselves. There might be non-DNA or RNA characters inside there. What we're doing here is putting as the input is the multiple sequence alignment, the model of interest. The prefix is -T3 so we can differentiate the different files from each other. And we put in a random sees 2, and we should change to 2 to a different value every time we run the program. So what we get from this is optimized parameters. So it talks about 4 gamma categories. The base frequencies vary for each particular base. Substitution rates, probabilities again are higher, and you can guess which are the transitions versus transversions. And then we see the AIC and AICC scores and BIC scores are all there. And what we get from this is 6 files, including the log file. We also get the best tree file, and we're going to use that when we're in R. We also contain this contains best T with 2 T's. This contains the NUIC file in this format. So we see our sample, a colon, and then the distance associated with that external branch. There's our other sample. There's another sample in its branch. There we go. There's the distance differentiating those different samples, and another sample. So you can actually construct the tree from the NUIC file alone. So what we see next is that we can load this in R on the server, install ape, install ggtree, read in our XML tree, and use the tools that we used earlier. So we can use ggtree to construct a phylogeny, something like this, for example. As you can see there, 2 and 3 are closely related. 4, 5, and 6 are somewhat closely related, and quite divergent are 7 and 8. So you see, yeah, we're aligning the tip labels there, so we can see them more easily. We can then do the same task of XML tree construction, this time with Bootstrap. So you see we're putting in the command Bootstrap there, and that tests the consistency of the tree. We use a different prefix so we can record that. And we'll see this kind of output at the end. That's the actual command. So you can see all the different things. So we're talking about the support. This is the next command after the bootstraps, is to assess the support. So this is the support function. For this particular tree, we're looking at the bootstrap trees. That's the file, and this is the new prefix we're using. So now we'll get this got a NUIC file, and what we can do is construct this with XML. You can see our bootstraps in there inside this file. So again, we load this with R, we visualize our tree. We're including the node labels too. And what we'll see is something like this. So there's our 83 we had before, and we can see how robust the different splits are. So we see that most of them are very robust, except for this split between this topology of 4, 5, and 6. Maybe there's a bit of variation there. 4, 5, 6, and this node down there. There's a bit of variation there. And then what we can do then is function all, and including the bootstrap metric. optimize the number of bootstraps implemented. So we can use our final observation is that we can combine all these functions together by specifying all. This is our MSA input, our model, our prefix, and the BS matrix metric at the end indicates that we want to do all the different options to optimize the number of bootstraps that are present to test the consistency of our tree sufficiently robustly. So we can go back to our directory we had before, where we have our sequences.faz file, and we also have china.faz file we can test as well. So I'm just going to zoom in so you can see that more easily. We can take those commands we had earlier and use them. So we know that raxml ng is installed here. So if we type in raxml, so we can copy raxml ng over like that. So what we can do is double check that that worked. That seems to have worked. And then we can try our first command here. So we're using that executable in our directory using the check function. And we're doing a multiple sequence alignment. So we give that input file as just seq.faz. Our model here is going to be GTR plus gamma. And we're using the prefix. We're going to call our prefix to be S1. So we run that, and what we see is that we have several outputs, including our log, which means that the alignment can be successfully read by raxml ng. What we can do next then is actually align our sequences. So the same sort of command structure, raxml ng dash msa seq.faz dash model gtr plus gamma dash prefix. Let's call it S2 dash seed. Let's call it some random numbers and let that run. So what we see now for the S2 files, we got 6 files that are produced, including the best tree. So if you look at the best tree using more, we can see that it's in Newick format, which means that we can write in with R quite easily. So what we can do is module load R to get the most recent version of R. So it's R version 4.2.2. We install ape if we haven't done so already. And see if that installs. And hopefully that will work okay. What we can do is check if that ape was installed properly. And then also do the same for GG3. So it might take a minute for these things to install, but that looks good. It's just reading in the different parameters that are required to install that library effectively. And then we can read in our best tree. So we see that we can't open the file that's of interest. So we can check here in Phylogen folder, we called it, we have to make sure that it corresponds to the correct name. So you see the stem is the same. That's dot best tree, but it's S2 dot raxml lowercase. And then we can know that our tree one is being read in okay, because we can see it there. So what we can then do is try that example command I gave you before, and we can see there where we've recapitulated that result, showing the divergence between those different FMDV VB1 sequences. So what we can do next then is do the same with bootstrapping. So we can quit and save this workspace. And we do the same with bootstrapping. We have the T7 output. And as mentioned before, what we can do is look at the support for using this bootstraps file. That's our key file from this step that creates the bootstrap file. And these are contained all the different trees that we want to look at in our alignment. So what we're going to do with the correct command is use the support command to take in our create a tree from the previous step called T3 S2 take in our existing good tree, S2, and then our parameter is --bs trees, and our bootstrap file is the one we had before, so it's t7.raxml.bootstraps. And a prefix for the output is going to be, let's call it S3. So let's execute that command. That is appear to execute OK. And what we can do next is then read in the this s3.raxml.support file. So let's copy that. We'll open R again. We need to restart GG3 and ape. Oh, here. And then we can read in that file. So that was called s3.raxml.support. We can check that the tree one was read in okay. And we see that the node labels are Canadian bootstraps. So when we visualize this, we'll see that those labels, oh, sugar, those are contained here. So now we've visualized our tree. A final one would be then to reapply this on the China sequences. So if we do that, we can just modify, oh, need to quit this part. Oh dear. We're going to use C1 as a prefix. We're going to check that the sequences are okay. First of all, China, that runs okay. It's good to go. We can then perform our alignment again using what did we call that? C1. So we're going to call that C2 as the prefix. We're going to change our input there. This may take some time because there's a large number of sequences there, so you can see that takes additional time to complete. So what we can do then is just be patient and wait for it to complete.